Good afternoon. Thank you guys for coming. I'm Jeremy DeSilva, for those of you that have not taken class with me yet. Assistant professor here at BU in the anthropology department. And this is the first of what hopefully will be many dialogues in biological anthropology that we want to have to really facilitate and encourage conversations around controversial topics in biological anthropology and see what kind of common ground can be reached. I'm very pleased to be joined by an amazingly distinguished panel of researchers, paleoanthropologists and biological anthropologists to talk about the Hobbit, a small female skeleton found in 2004 of a remarkably small individual with a remarkably small brain. But first, let me introduce the panel. On the far left, professor of anthropology at BU, this is Matt Cartmill. Right to his right is Bob Martin. Dr. Martin is the curator of biological anthropology at the Field Museum in Chicago, and he has a particular expertise in brain evolution and scaling issues in biology, which are both incredibly relevant in trying to understand the Flores hominid. And finally, on my immediate left is Dr. Fred Smith. Fred is the department chair of the Sociology and Anthropology Department at Illinois State University and has a particular expertise in Pleistocene fossil hominids and modern human origins. We're here today to talk about a very special little lady, again, who was discovered in 2004. Her discovery led to the erection of a new fossil hominid, Homo floresiensis. She was unusual in how recently she lived. She lived only 18,000 years ago, and she was very unusual in her small body size and her amazingly small brain size. Now, because of this unique pattern or this unique morphological set that she has, there are some researchers who have suggested that instead of her being a new species, she's in fact a modern Homo sapiens that suffers from a pathology known as microcephaly. And that debate has actually gone on for the last 5 years or so. And today's session is to fill you in on what the most recent literature is, most recent information is, and to have a discussion regarding this so-called controversy. First, I want to recognize Kay Brown and her work in organizing the dialogues of biological anthropology. And again, this is the first of what I suspect and hope will be many of these. And what we'll do is we'll essentially have a conversation up here for 15, 20 minutes or so, and then we're going to open up to questions from yourselves. When you get an opportunity to ask a question, I just ask that you wait until you have a microphone so that your voice can be recorded for the video. And with that, I want to throw the first question actually Fred's way. I've been talking about this small female skeleton, this LB1, Liang Bua 1, and yet she's not alone, is she? There were other individuals that were discovered in this cave.
Yeah, I think it's important to stress that fact. There are 97 skeletal elements from Liang Bua, but 2/3 of those are Liang Bua 1. So I think 61 are Liang Bua 1. Then Liang Bua 6 has 14 or 15 elements. So we actually know a fair amount about Liang Bua 6 as well. And what we know about Liang Bua 6 pretty well mirrors what we have for Liang Bua 1. In other words, it's an individual of very small stature. It has a very small mandible, no skull. But what we know from LB6 pretty much reinforces what we know from LB1. But all of the other individuals are known from no more than 3 elements, and many of them are known from a single element, perhaps only a single tooth.
But they're all small.
They're all small. There may be one individual in which we could question if this individual is as small as the others, because this is a piece of radius that isn't complete, and it's not clear how much longer it would have been. But yes, I would be willing to bet big bucks that all of these individuals are very small body size.
So, Bob, the problem though is not the stature of these individuals, because we have pygmy humans today. We have people who are small today. So the issue is not the stature, right? It's the small size of the brain.
No, that's exactly the point, is that having a small body even in modern humans, is not a big problem. And in fact, in that region in Southeast Asia, there are small-bodied human populations. And on Flores and other islands, there are what we call pygmoid skeletons from archaeological excavations. So small body size is not the problem. It's the small brain size, this grapefruit-sized brain. And I would emphasize that we only have one skull. We may have fragments attributed to other individuals, but we only have one skull. And so there have been comments, even in scientific papers, referring to a small-brained population of hominids, and we do not have that. We have one small-brained hominid and some fragments. And I'd also say, I mean, it looks as though the other individuals were small, but if you just have an isolated premolar tooth dated at 95,000, does that really tell us that individuals as small as that were present? And Fred has mentioned the radius. So certainly with some individuals, they were clearly small, particularly the individual with the second lower jaw or mandible. But, I'm not convinced they were all that small.
It might be worthwhile pointing to the cast of the Liang Bua specimen there so everybody knows what we're talking about
This
when
is
we
it.
say small.
This is the Liang Bua 1 specimen. And to give you a context, this is a fairly normal modern human. So, I mean, you can see how small that brain case is.
And 18,000 years ago, modern humans had evolved.
Oh,
Right?
absolutely.
So we're not talking about a possibility that modern humans are not on the scene yet.
No, in fact, this is the only thing that I'm aware of, okay, that, as Bob pointed out in his work on the brain size, that substantially deviates from what is a fundamentally modern human skeletal package that's been around everywhere for the last 30,000 years. Mm-hmm. So, I mean, that's one of the things that's really unique about this specimen is that it is so unique. I mean, there's nothing else like it, and it was completely unexpected. I mean, when I first saw the manuscript on this, my first thought was they've got the dates wrong. The dating is wrong, because this sort of something like this at 18,000 years ago.
So it should
It
be
makes no sense.
3 million years old and living in Africa.
Well, I don't know about 3 million years ago, because that face is not doesn't look like either the two primitive forms of Afarensis or Tim White. I mean, it looks very different from both of those.
Now, Bob, though, when you first read the manuscript, you thought there's something wrong with this brain.
That's right.
Yeah,
From the brain size.
So what is the problem with having a hominid with a brain that small? Why can't we have a hominid of that age with a brain that small?
Well, the first thing is, having worked for 40 years on brain size throughout primates, I just knew it didn't fit anything I'd ever seen before. And Fred referred to a curve. When you plot every single cranial capacity of every hominid, fossil hominid we know, you get a relatively clear curve, and it swings upwards as we get close to the modern day. The only fossil that does not fit on that curve is this thing. It falls way below anything we have for 18,000. That brain size 3.5 million years ago would be acceptable.
But probably not that total morphological pattern of the
Oh,
skull.
absolutely not. I agree with you.
That would stick out.
I'm just talking about brain size, but
Right.
if you look at the face, that's clearly Homo to me. But
Yeah.
So the original explanation of the authors was that this was an island dwarfism effect, that something like this individual here, a Homo erectus from Indonesia, from Java, was somehow, the face is gone.
Oh.
There you go.
Face broke off. in the face.
I don't recognize that specimen.
This population somehow got isolated on the island of Flores and underwent a known biological phenomenon, island dwarfism, where the body reduced. So why not that as an explanation? What is the problem with that as an explanation?
yeah, the first thing is that there is a so-called island rule, which Van Balen originally suggested, I think, in 1974. But the idea was that mammals living on islands often get smaller. But it's about body size, and nobody ever looked at brain size. Nobody has ever said there's an island rule that says your brain gets smaller. And so what I worked upon is standard relationships that we have been studied across mammals in the reduction of brain size with body size. So if you start off with a modern human this size and try to scale that down, you would have to have a body height of about 6 inches to get a brain that small. This is what the general database tells me.
Yeah, that's a real problem, to have a hominid 6 inches tall.
6 inches tall with a skull 5 inches tall.
Not quite, but yeah.
So the island dwarfism explanation seems to be losing steam. The data doesn't support it, and even the authors.
They changed their mind within a year.
So what is the latest explanation then for the brain size?
Their explanation is that what we have with these with something like this is that this is the remnant of an even earlier migration of humans out of Africa before the Homo erectus migration. So the similarity would be between something like this. This is KNMER 1813, which is about around 1.8 million years old, 1.9 million years old. And so that something like this guy came out of a line something like this. So they were small to begin with, and they just remained small. And one of the reasons why they made this argument, as Bob alluded to, island dwarfism clearly is a phenomenon that relates to body size reduction, not brain size reduction. So this fell out. This was an anomaly within an argument for island dwarfism. It didn't fit. So, and the other thing was that the limb proportions here were very primitive limb proportions, and there was no evidence that dwarfism changed the limb proportions to make things look more primitive or to have a more primitive condition. So it became more parsimonious to say, well, they didn't really reduce, they just started out this way and got to Flores and stayed that way. But again, the big problem is where's the rest of the evidence for that migration?
Right, there's a large geographical region, a large time scale with a limited fossil record supporting that connection. Now, Bob, you have a different take on this, that this is not a remnant of an early Homo expedition out of Africa that made it to Flores. Instead, this is a pathological modern human specimen. Can you?
Well, the first thing is that curve that I mentioned shows that in all other hominids, brain size increased over time. So we have to explain why it didn't in this one hominid. Secondly, as Fred has pointed out, there are no intermediates. So there's nothing between Africa and Flores to document this hypothetical migration. So basically what people have been doing, first of all, they tried island dwarfism as an explanation for the small brain, and when that didn't work, they're now saying that it is an early surviving hominid. And I don't think that's any more convincing, and particularly because of the stone tools. You know, I mean, the stone tools were initially described as quite sophisticated, and I talked to a lot of colleagues who are biological anthropologists, and you say, what about the stone tools? Well, you know, they're probably not associated or something. The original discoverers said they were associated, and this Hobbit made stone tools like this.
So can you explain to them what do you mean by a sophisticated stone tool? What were these guys doing that was perhaps beyond the mental capacity that you see here?
Right, it's a fairly heterogeneous collection of stone tools, but some of them are clearly made with what is known as the prepared core technique, and you can see it in this. This is a flake from a prepared core, and on this side you can see scars where material has been removed to make a particular shape of the core to start with, and then you make a striking platform, and then you use an implement here to hit at precisely that place, and that gives you this flake. And there is a percussion bulb on the other side, which is always present. So it's pretty easy to recognize this as a flake from a prepared core. And in fact, there's a scar on here. It's possible that a previous flake was removed here. And so prepared cores and flakes were not used systematically by any other hominid except Neanderthals and Homo sapiens. Homo erectus didn't do this, except very occasionally if I've not seen any good evidence that Homo erectus did this, certainly not until later stages anyway. And so there's this, and there are the cores in the collection. So you can see radial cores that were clearly prepared and then had flakes struck off of them. And the second thing is that Neanderthals tended to make rather broad flakes. And so with Homo sapiens, you get what is called a blade, which is simply defined as being longer than broad. If it's broader than long, then that's what Neanderthals were producing. But blades, and particularly long, narrow blades, and there are some from Liang Bua that are this size, if not bigger, and they're not much wider than this. So you've got definite blades in there. And usually with Homo sapiens, blades are made to be bound to a piece of wood of some kind, either to make an arrowhead or to make a spearhead. And so if blades were being made, and some of them have been retouched, all of this tells me this is a pretty advanced stone tool culture that we're seeing there in the cave.
Bob, one of the points you made in the webcast that we did before we came over here this afternoon was that modern humans, anatomically modern humans, people looking just like or pretty close to like the people living in these areas today, had in fact moved into the Malay Archipelago and all the way over to Australia by something on the order of 40,000 years ago, long before this specimen is found at Liang Bua. So is there any reason for believing that the Liang Bua population, whatever it represents, were the only occupants of these islands in the Flores Strait at this time horizon?
That's a very good question. The key point is that Mike Morwood, who leads the archaeological team that excavated that skeleton, argues that Homo sapiens never reached Flores until after the Hobbit died out. And so he says the only possible maker of these stone tools was the species Homo floresiensis. There's an ash layer, I guess around 11, 12,000.
The ash layer is about 16 to 17.
Okay, there was a huge volcanic eruption that...
Right after the Hobbit.
Yeah.
That's 18,000, and so soon after that there is an ash layer that... and anything beneath that, Mike Morwood says, is Homo floresiensis, and then above it you get stone tools that I can't distinguish from these, which are attributed to Homo sapiens.
The only difference that I would add there is that when you do get these later complexes after 11,000 years, they do have some additional different tools than aren't there earlier. In other words, they're using more groundstone and other resources. All of this stuff is made on fairly low quality material, and after 11,000 years ago, they're using more cherts and things like that. So there is a greater diversity of tools and a little bit greater sophistication to those tools at around 11,000.
One of the problems with stone tool making is that if the material you're using is not very good quality, it's difficult to make something this good. And so I think you're right, different materials were being used, but I don't see any significant change in the technique.
Well, there is some significant change in the technique because there are new types of tools that are coming in that weren't being made prior to that. Such as they've got these bifacially flaked what do they call them, scythe-like tools and so forth. But now these tools are tools like that are continuing to be made. And I mean, again, I agree with what you said, that raw materials clearly are going to affect what the tools are going to be like. But those cherts were there when the Flores people were making the stuff out of the other material, and one wonders why they didn't use the chert. But I'm not really disagreeing with you.
I think you're right. I mean, this whole thing needs to be properly re-examined, but it's quite possible that Homo sapiens got into Flores and was making tools
Absolutely.
like this, and there was a pathological individual, then this eruption wiped all of the Homo sapiens out, and then a new wave of invasion had different stone tool making techniques. That's a possible
certainly
scenario.
possible.
So I want to stay on the dating and the first appearance of Homo sapiens in this region for a moment, because some of the earliest specimens that have been attributed to Homo floresiensis are dated at 74,000.
Right.
Is that
at
right?
least. Well,
And
there's
that's prior to what has been suggested for the first
The
appearance for
premolar
Homo
is
sapiens.
95,000.
maybe 90.
So we go 95,000. So is there any evidence that some of those earlier specimens can be lumped into this Homo floresiensis category? Are they all small? Is there any morphological reason to suggest that these are not just late occurring Homo erectus? So who are they? Essentially is what I'm asking, and can we know that?
First of all, there's very little fossil evidence that relates to the appearance of early modern humans in that area. There are 2 or 3 specimens. The only one that is anywhere close to this age is a skull from a cave in Borneo called the Naya cave skull, the deep skull from Naya, which is a subadult individual, probably a 14, 15-year-old partial skull. It's small, but it's nowhere near this small. I mean, you don't bat an eye in terms of looking at this specimen and saying it's a fully modern individual with no problems.
And what age?
Well, that's the big problem. There's been a lot of debate about the dating of Naya Cave. However, there's some recent evidence which would suggest that deep skull is about 40,000 years old, and I think that's the basis of this argument. When you get to Australia, there are 2 basic arguments about when modern people got to Australia. It's based on the same evidence. There's no skeletal material that's this old, but there are dates for a series of early sites with tools in Arnhem Land and so forth. And the debate is which technique gives you the best dates. There's one technique, what is it called? Is it obsidian hydration that gives the 60,000 year dates? And so one group says, okay, that's the earliest date for modern people to get to Australia. And other people say, no, you can't trust obsidian hydration. We're going to take the AMS radiocarbon dates that say 40,000 years. So it's either 60 or 40, depending upon which dating technique. It's the same evidence. It's just the question of, okay, are you going to accept these obsidian hydration dates or not?
I'm puzzled by the fact that modern humans are crossing the substantial sea gaps in Wallace's Line and making it all the way to Australia, even during periods of glacial advance and sea level fall, and they never made it across what would it have been at a minimum 20-kilometer gap between the Lombok Straits over to Flores and the chain of islands beyond Flores. It's not just Flores, right? Flores is hooked up with Sahelia, isn't it?
There is also a gap between them too, I think.
Okay, so that's the difference between Wallace's Line and
Liedecker's Line.
Liedecker's Line, yeah.
So just for clarification, so that people know what we're talking about, why is it so hard to get to Flores in Australia?
Because you have to cross open sea.
Yeah.
There was no time that Australia and New Guinea, or at least within the time we're dealing with humans, okay, that Australia and New Guinea was connected to Southeast Asia. Many of the islands like Java we were talking about, or Borneo, for example, they, in low sea level, they were part of something called Sundaland. So that was all dry land there. And New Guinea and Australia were also joined together. Tasmania was joined to this. But these islands in between, the Lesser Sundas and some of the Philippines and so forth, were never connected to either one of those things. So to get from Southeast Asia to Australasia, you would have had to have crossed some open water barriers, even at low sea level.
And yet we know that happened by a million years ago. There are stone tools on Flores that are
If those
dated
dates are
that
correct.
age,
And
yeah.
those dates, the dates for these early stone tools, I mean, again, we weren't able to talk about all the problems today, but those dates are not accepted by everyone. The dates are probably accurate. Whether they are actually dating those artifacts, that's another question.
You know, one of the things that has bothered me from the very beginning of these discoveries on Flores is the question of how you go about crossing substantial oceanic gaps if you're Homo erectus. Now, some people like to think that they had seagoing vessels. I'm pretty skeptical about that.
Right.
The usual line of goods that you have gotten from zoogeographers ever since the 1930s is that animals are somehow clinging to floating masses of vegetation, which get swept out to sea by storms, and then the animals on them get off when the floating mass of vegetation docks at the next island. And I have always had two problems with that. One is I, at least personally, have never been able to find an example that anybody has ever found of a mammal clinging to a floating mass of vegetation out in the middle of the Atlantic or in the Sunda Straits or anything else, despite the fact that storms continue. And secondly, it has to be a pregnant female. You can't. It doesn't matter if you wash a male Homo erectus to Flores. And just one pregnant female is going to give you some of the most terrible founder effect sampling errors. And,
I don't-
You know, it's
Maybe we don't even want to go there, Matt.
well, I mean, we have to. You get one pregnant female, and so her offspring and her become the only genetic material that's the basis for the subsequent population. And all right, so yeah, I know there are some dental pathologies or abnormalities on this specimen, and sub rosa jokes here are being made about inbreeding, but it's true that if you don't have any more genetic variability than that, then I would expect that in a situation like that where the population size is extremely limited, you're going to have a hard time over the long run maintaining a genetically viable population.
Absolutely. Absolutely. And that's from my standpoint, one of the big problems I have with thinking these guys got to that island a million years ago. Nobody else ever came. And there was a viable enough population established that could exist for that long a period of time in isolation.
So what about the other fauna that's on the island? I mean, it's not just Homo erectus or floresiensis or sapiens that made it to the island, whatever we're going to call this thing. There are stegodons and
There are stegodons. There's not modern genus Elephas, but a closely related Pliocene elephant genus, and this is a dwarf variant. So it's another example, supposedly, of this supposedly widespread phenomenon of insular dwarfism. And it is true that in the Pleistocene you get, in a number of places, including the California Channel Islands and various Mediterranean islands, as well as Flores, you get these dwarf elephants that have presumably been swept to the island by storms or other kinds of natural catastrophes. But the interesting fact about elephants is that if you drop an elephant in the ocean,
He will swim.
not only will he swim, he will be able to breathe by sticking his nose up above the water and keep that up for long periods of time. So a lot of these insular faunas that have managed to colonize islands tend to be some. Fred's looking for evidence of a trunk. Tend to be animals that can sustain and can withstand prolonged periods of immersion in seawater, one way or another. Either they've got low metabolic rates, like the Komodo dragons that colonized Flores, or they're fairly large in body size, like elephants. I've always had a problem with Microcephaly being rafted across, the Straits of Madagascar to colonize, start the Madagascar lemur radiation, and I have a problem with Homo erectus and tsunamis and clinging to floating rafts of vegetation for weeks on end while being pregnant.
Go ahead.
This is a very good point that you've made, and so it's very important to make it clear that Mike Morwood and his colleagues are arguing that this all happened exclusively on Flores, that an early hominid got to Flores around 1 million years ago now, that we're told, and independently developed these sophisticated stone tool techniques from nothing, and that there was never any migration inward or outwards. And so this is a purely Flores story. And to follow up on your question, I mean, there are special creatures on these islands, such as stegodon dwarf elephants, and there are things that get bigger on islands, like the Komodo dragon. Exactly. But as far as I know, apart from the Hobbit, there is no other species on Flores that is unique to Flores. And so stegodon is something that's distributed throughout the region, and large lizards are spread out here. I don't know of a single other species, and particularly not a mammal species, that is unique to Flores. And so why should this happen just with that one hominid?
So you would expect that if this is a real population, that we would start finding the remains of late Pleistocene small-bodied, small-brained individuals on some of the other islands of the Indonesian
That's
archipelago.
what I would expect, and I think Matt's point is a very good one. You can calculate what the likely population size would have been of an early hominid of our body, or that body size on Flores. And I don't think that that is viable over time without inward migration. I think without gene exchange, there is no chance whatsoever that a hominid population would survive and evolve on Flores without any interaction with any other landmass.
I wouldn't say it's impossible, but I would say that it strains belief that it could happen.
Yeah, there's an interesting sort of interactive study incorporating social anthropological and population biological and archaeological data that are a series of studies, literature that's been emerging over the course of the past year, which suggests that in the absence of exchange, culture tends to evaporate. That when you look at populations that are either geographically isolated at the edge of other populations, or that are part of human populations with very low population density, you can see over the course of time that they get more and more impoverished in terms of their technology. For example, if you trace the radiation from west to east across the Pacific of the Polynesian migrations, the cultural repertoire that people started out with in the initial departure from the Indonesian area, and the cultural repertoire that they wound up with on the extreme western edge, there's quite a lot of difference. As you move across the Pacific, the technologies get fewer and simpler. The reason for this apparently is that if you live in a small village or tribal kind of community, there are likely to be a very small number of people that know how to exercise certain skills. And if those people die accidentally by whatever means before they have managed to pass those skills along to somebody else, then those skills disappear from that population forever afterwards. Now, in light of that, it seems to me correspondingly unlikely that we're going to see Flores as a locus for the development of innovative techniques. It's not impossible, because the most western point in the Polynesian radiation is Easter Island, and those people invented writing and did a number of other interesting things that are not seen elsewhere in Polynesia. But it seems to me correspondingly improbable, and there's a developing sort of statistical literature that is able to assess the probability of innovations given certain conditions of isolation.
But whatever the issues may be, the fact is that we do have these very small-bodied people with very unusual limb proportions, okay? That, can we say there's no chance that was due to pathology? Well, no, we can't say that for certain, but we can say that those differences in limb proportion and that elongated foot and the characteristics there are not related to their small body size. So we still, if we get away from just a minute from the small brain, we've still got the issue of explaining what's going on with these people. Forgetting the small brain for a minute. I mean, it's a big problem. But there are other things about these guys that also have to be explained as well.
I was about to say we haven't gotten to the feet yet.
Yeah.
Feet are
No, it's a
fascinating.
It's a very pedestrian topic.
Fair enough. Yeah, the postcranial anatomy is odd of this individual, and it seems to me that to address that question, we need to know a lot more about the postcranial skeleton of people that have microcephaly and some other diseases that may explain this, and yet there's nothing. We don't know anything about microcephalic postcranial skeletons, or
There's
very little?
there is clinical literature that shows that the genes that produce the small brain and microcephaly also affect dental development and also affect the postcranial skeleton. The pelvis does not develop normally and the thigh bone doesn't. So we know that these genes also affect the postcranial skeleton. But the problem is that I can find at least 100 skulls of modern human microcephalics in museums around the world, and I've looked at about 25 so far. But if you look through the catalogues, they're there. I have not been able to find a single postcranial skeleton of an individual that survived to adulthood, because the thing we shouldn't forget is that the LB1 skeleton is from an adult. All 3 molar teeth are present, so it was clearly adult. And so what I'm looking for is a postcranial skeleton of a modern human microcephalic in a museum collection, and I can't find one.
Yeah.
Maybe it's out there somewhere, but I haven't yet been able to find one. I'm in touch with a doctor in Zurich who is a specialist on microcephaly and trying to get a CAT scan of modern human microcephalic patient, but the chance and the ethics of that are questionable. So I don't know whether this is going to happen, but you see, the thing that really gets to me is that people can publish these papers saying that the postcranial skeleton cannot be pathological when nobody has checked. That I find unacceptable. How do the reviewers let this through? You just say this cannot be pathology. Well, how do we know?
Well, I don't think we do know for certain. I agree with you there. I think I will say that if I were a betting person, I really would bet that the postcranial anatomy of these things is not pathological. But I certainly agree that it's one thing to say that, it's another thing to have the data that show that is indeed the case.
Yeah, Jakob and his co-authors early on in the history of the literature on this specimen argued that it was pathological because it was because the postcranium was asymmetrical in a way that you could not find in a modern non-pathological skeleton. And I think that one of the major contributions to this literature was Bill Jungers and other people at Stony Brook and his collaborators going through and showing that, in fact, the degree of asymmetry in the skeleton was not something extraordinary for a modern human population of people with normal limb bones.
Yeah, and I'll add to that as well that aspects of the postcranial skeleton, in many ways, there's a publication on the wrist, publications on the shoulder, and now the foot that seem to suggest that some of the postcrania ends up looking quite a bit like earlier hominids, and so it would end up being a pathology that mimics something that you see previously in time, which certainly could be the case and could have happened. But it is interesting evidence, nonetheless, from the postcrania that the wrist morphology and foot morphology end up looking more ape-like and more like, in certain ways, Australopithecus. So clearly there are questions and issues to sort out, and much more research to be done by these folks, and a lot more dialogue to continue. I think now I want to open up the floor to questions from you guys. Because hopefully there are a bunch, because we have plenty of questions about this, and hopefully you do as well, and maybe we'll have some answers or more questions. So Kay has the microphone, and who wants to start us off?
Do not be shy.
So I don't know much about this topic at all, but I was just wondering if from the skeleton you can tell if the adult female had had offspring. Is that
No.
possible?
No, not because even if you, the only way you can do that if you have certain parts of the pelvic girdle where you can see the impact of the birth process on the ligaments that hold the pelvic girdle together, okay? And particularly you can see this on the pubic bones that come together in front. Those parts are not here, and so it wouldn't be possible from that perspective to say one way or another. And let me hasten to point out that oftentimes you can have pelvis of females who have given birth that don't necessarily show those what are called parturition scars, so they're not a 100% indicator. And sometimes you can get parturition scars if you have some other kinds of stress on the pelvic girdle too, so it's not a 100% indicator. But it doesn't really matter in this case because the right parts aren't there.
Can I just point out the microphone is not to make things louder for us, it's for the tape recording.
Right.
So don't be worried if it doesn't seem to be resonating in the room. It's not supposed to.
Yeah.
I don't really know much about this either, but can you tell from the teeth what kind of diet they would have since they have these tools? Are they hunting game? Are they not? I don't know. What's going on with that?
Do you want to? And I'll go off.
It is possible to get an idea about diet from looking at patterns of microwear on the teeth. I'm not aware that they have actually done that. So now, if you look at the material in the cave, there is indications of the use of fire. Some of the juvenile stegodon bones, the subadult stegodon bones, show evidence of cut marks. So the inference is that these guys and gals had a fairly broad-based diet, what I would call the Pleistocene Homo diet, which is you eat anything that you can eat. Okay.
But the teeth are fairly heavily worn.
They are
so
heavily worn.
Whatever it was abrading the teeth.
Yeah.
Nobody has looked, correct me if I'm wrong, you guys, nobody has looked at striae of Retzius and so on, at perikymata in the teeth to see if these things are showing a modern human sort of pattern of dental eruption and development, have they?
If they have, it's not published, and I have no knowledge of it at all.
Would you need a juvenile mandible for that? Anyway?
You'd need some juvenile teeth. I thought there were.
Are there?
I don't
in
know.
the collection?
I don't know.
Okay.
I don't know. I don't know that you would need necessarily juvenile teeth. You would need fairly unworn teeth to be able to, but.
Rena.
The current argument for, or explanation for the brain size is that they originated from like an earlier migration. Then how do they account for the other facial morphologies, which are more modern and human-like and less like an Australopith?
Okay. Well, the argument there is really that even though the limb proportions may be more like an Australopithecine, they're talking about them descending from something like this, which is supposed to be an early Homo. Let's call it Homo habilis, okay? And a lot of the anatomy of the face is not too terribly different from this. So they're not really arguing that this evolved from something like Lucy. They're arguing that it evolved from a later population of Australopithecines. And although we're not absolutely sure what the limb proportions of this group of early Homo was, there is one skeleton from Olduvai Gorge that's supposed to represent Homo habilis that has the the same kind of body proportions as Lucy does. So that's where it's supposed to come from, something like this. Okay, does that help?
And a key part of that argument then is that there is no evolutionary change in brain size for 2 million years, which is maybe it's true. I would just point out there is a problem. The postcranial skeleton is a weird mixture. And in many respects, the bones cluster with chimpanzees, not with other hominids. Okay, if you look at the relative length of the femur and you look at the foot bones and you look at the wrist, it looks more like a chimpanzee. And yet the pelvis seems to be clearly shaped like a human pelvis or hominid pelvis, starting with Australopithecus. So there's this strange mixture of features, and the thigh bone, the femur, has a carrying angle on it, so it clearly looks, and the foot, if you plot the individual bones, they seem to fall with chimpanzees, and yet it looks as though the big toe was not divergent. It was bound in with the other toes, so you've got a human foot, and yet the individual bones have measurements. And if you just look at the size of the foot relative to the lower leg is huge.
It's 70%.
Looks nothing like
of
a
the lower
hominid.
leg, yeah. So imagine a foot, 70% of the length of your tibia. It's a big foot. Yeah. And the toes are what make up most of the large foot. It's not a big foot. That's not what I was
So
saying.
that overall
Has a
pattern
large
doesn't fit
foot.
anything we know.
Yeah.
There is nothing, either an ape or a hominid, that looks like this. And so why not pathology?
Thank you for your talk. I just had a question about the lower premolars. If I'm not mistaken, it's missing one, if not both, of the mandibular premolars. And are there, is this correct?
I can't hear that much.
She's asking
Missing
about
the
the
lower
asymmetry of the
premolars.
lower premolars.
Yeah.
You mean the asymmetry or the roots of the premolars?
No,
Because
he means there's one too few premolars on one side.
I thought it was on both sides, but maybe I'm wrong. But I mean, certainly there is a congenital absence of at least one of the premolars, and I can't remember the details. I thought it was affected by. Yeah, I'm sure it affects both sides, and it looks there is an apparent asymmetry, but it's not due to the premolar. So I think a premolar is absent, congenitally absent on both sides. I think the X-rays show that. So, and in the upper jaw. This was described as a feature of the species Homo floresiensis. In the upper jaw, there's a premolar that's rotated 90 degrees in both tooth rows. And that is an anomaly that you see in people. Dentists see this from time to time in people, but it's not natural. It's an anomaly.
Well, Fred, you were saying that this appears sometimes in the Carpina and Neanderthal population.
Carpina Neanderthals, the lower premolars are often rotated. It's very interesting. It's a high frequency of that rotation.
But these are uppers.
Yeah, well, I mean, premolars tend to be a bit unstable anyway, and particularly the P4s do. And I'm looking at this, and the reason I'm not sure is that you have to understand the cast we have of Liang Bua 1 is a cast from an organization called Bone Clones, and these are not really casts, they're sort of models. Okay, so I'm not sure, but when I look at this, it kind of looks to me like, let's see, too, that there is, that the P4 on the right side is gone, but not on the left side.
There's the socket.
Yeah, there's the socket for P4, that's P3, there's the canines. So, looks to me like it's one side, but
It depends
As I
what these
recall,
are.
that was one of the things that Eckhart and company and Jakob pointed to in talking about the asymmetry of the
they actually
of the skeleton.
they looked a lot at the facial asymmetry too, and some other papers have first of all argued that the facial asymmetry is not out of the range of variation of normal humans, and they've also pointed to a phenomenon that happens a lot in certain groups that's called, I always forget this, phagocephaly, something like that, where Plagocephaly, that's it. Where the head, where the baby is laid in a certain position a lot, and while it's developing, and this causes a flattening in the occipital in that area, and it has concomitant effects on the face. And I'm very familiar with this because my first work in physical anthropology was working with Native American material from the American Southeast, and these were often cradle boarded, so they were bound to a board. And I mean, you talk about a bunch of people with flat backs of their heads, and it has all kinds of impacts on the face and facial asymmetry. So what they're really arguing is that it's not a pathological kind of
So the Hobbit invented cradle boarding as well.
But the original argument of asymmetry in this skull is there's a recent publication suggesting it's not outside the range of variation you see in modern humans, modern chimps, modern gorillas. So that argument is probably not
real convincing.
Not real convincing. Okay. Other questions? Caitlin? And then Rachel? Oh, Rachel, then Caitlin.
So I'm just curious as to how plausible it is that Homo habilis got all the way to Flores when diminutive was surprising to people when it was Homo erectus getting there. So how could they is there any proof at all that Homo habilis could go all the way there?
No. There is no proof. But on the other hand, you have to understand that if this was the first migration out of Africa, it would have been a wide open niche. You're still moving through what is essentially a tropical environment. Okay, there would have been no close competitors to them, so they could have potentially spread very quickly. But that's a theoretical argument. You're asking me if there's any proof that they did it, and outside of that head right there, and the arguments with the postcrania, and maybe this new genetic sequence from this pinky from the middle of southern Siberia, there is no evidence.
I think there's one thing to be said in favor of it, and that is that early Homo, that everybody agrees is early Homo, shows up pretty much simultaneously in East Africa, the Georgian Republic, and Java. Okay, at somewhere between 1.9 and 1.7 million years ago, if you buy the older dates for the earliest material from Java. And while it's possible that they just sort of bolted and ran for Java in the course of 200,000 years after first leaving Africa, that distribution, those dates, and the fact that we simply do not have any fossil record of hominids in between those three places suggest that there may be a whole bunch of stuff going on in Africa that we are not yet picking up in the paleontological record.
She asked for
I said
proof.
Africa, I meant Asia.
She asked for proof.
But the thing is, it has been argued, this is one of the diminutive skulls, and there are 5 known now, and we have cranial capacities with 4 of them have been published. And as you can see, the brain is relatively small. And so it's been argued that this actually would be an intermediate between Africa, and this is said to be close to Homo ergaster in Africa. So this could be an early member of Homo that got out of Africa and eventually led to the Hobbit. This is the argument that's been put forward. The trouble is this is 1.7 million years old, and that brain is 50% bigger on average than this. So that you still need a brain size decrease. And these guys were quite small, but not that small. And so
and not only that, these are too late to have been that pre-erectus movement.
Right.
Yeah.
But this is 1.7 million years old. But some people would argue that that is your fossil evidence of an intermediate. I'm just being fair here. I don't know why.
And then wouldn't there be evidence somewhere between Flores and Africa that there was an earlier radiation of Homo out of Africa?
You have to understand that any time you're dealing with paleoanthropology, the fact that there might be evidence out there is always a possibility, but you got to find it. And these things These things don't just jump up when you're flying over in an airplane and say, here I am. I mean, it's hard to find these things. So the fact about the matter is there may be all kinds of evidence out there that we have not recovered yet. And just like Bob and I in discussing about this is that what we really think about the whole thing is that we shouldn't be excluding explanations when we have so little data. So what Bob is reacting to, and I agree with him 100%, is that, you know, they're saying, well, this thing cannot be pathological. I don't think we can say that. We don't have enough data to say that, so we have to keep an open mind. But I agree with you. I wish I had more. I would feel more comfortable if there was more evidence about this pre-erectus radiation. But right now, we don't have it.
Are there Pleistocene sites in India and
yeah.
places like that would, those would be sort of the places where you would see one of these migrations?
Fred, correct me if I'm wrong, but I went to India to look at Cretaceous deposits, but in a couple of places I was walking over stone tools.
Oh, yeah.
I was walking over stone tools like this.
Well, our stone tools like this.
Hundreds of them. And unlucky I didn't find anything that nice, but I was walking over obvious stone tools. As far as I'm aware, there is one skull fragment for the entire continent
The
of
Narmada
India.
skull.
Armada, but that's much later.
And but that's all that we have as a hominid,
That's correct.
as a fossil hominid
That's
for
correct.
the entire subcontinent. And yet,
correct.
that is the only way Homo erectus presumably could have got from Africa
Sure.
to Southeast Asia.
And so the absence of this fossil evidence is not.
So I would never argue this because I think we have huge gaps in the fossil record, and so we have to find it. But the short answer to that is no, we don't have any evidence. It might be dug up tomorrow, but we don't.
We don't
today.
know.
Keelan.
I was just wondering, what do we know about the cognitive abilities and or handicaps of modern microcephalics? And what does that tell us about the possibility of the stone tools being associated with these Hobbit individuals?
Okay, I mean, the first thing is that this is associated with stone tools like that, and but that doesn't tell us that this individual made those stone tools. And if I'm right, this was a pathological individual, and there were other individuals in the group that had normal-sized brains that were making these stone tools. But the thing is that people tend to think that microcephaly is one condition. There are 400 different genes, and a mutation in any one of those genes can produce a brain a third of the normal size. And there are dozens of syndromes that have been recognized according to the different genes. And there's this huge variability in everything, including survival and including cognitive abilities. So if you just take the available museum specimens, there was a beautiful survey by Hoffman in 1984, and he gives data on brain size. And the average is 400 cc's for modern human microcephalics, exactly like this. And there's also a spread in ages of death. And at least 75% of people with microcephaly die before they reach adulthood. And I would say they're irrelevant because the Hobbit clearly was an adult. It had the adult dentition. But there's a small percentage of people with microcephaly who survive into adulthood and do incredibly well. And there's one case of this guy who eventually died at the age of 73 with a brain this size. And people we used to call midgets modern human dwarfs are people who have a brain more or less the same size as me, but a small body. And a midget is somebody who's in proportion, so a small head on a small body. And midgets were commonly seen in circuses and films and so forth, performing. And I was telling Matt earlier on, unfortunately this has been removed now, but there was a YouTube sequence showing a modern human microcephalic in India breakdancing and doing weightlifting and interviewing. He regularly appears on talk shows. And this guy is clearly, he has a special condition called MOPD2, and that is known to be compatible with survival. And so people can do remarkably well with some of these syndromes. Most of them are devastating, and people die before they reach sexual maturity.
Yeah, there's a lot of evidence along various lines that it is surprisingly possible to to have essentially normal human cognitive and mental capacities with a third of the normal human brain tissue volume. I was just looking at a paper yesterday that was published a couple of years ago on a French civil servant with hydrocephalus, and the French physicians had done CT scans, and hydrocephalus is a case where the cerebrospinal fluid builds up inside the brain and it compresses the brain tissue. And this guy had a layer of brain tissue about 1 centimeter thick on the inside of his skull. And he was, a French civil servant, and that isn't intended as a slur on the French civil service. He was of normal brain size. I have seen, incidentally, this and other similar cases referred to on the web by various kinds of spiritual and religious bloggers and web builders as proof that the intellect has nothing to do with the brain. And it resides instead in an immortal part that has no connection with mere material tissue. I think we have a lot of evidence involving things like lobotomies for believing, and brain injuries for believing that that isn't true. But nevertheless, it remains mysterious that there is less correlation than we would like to think between brain volume and intelligence, given this overall pattern throughout the Pleistocene of a slow, steady increase in human brain volume. Something's driving the increase. Exactly what, I think we really do not have a very firm grip on yet.
Oh, back corner.
I just had one question. So I know the feet are really big. Is there anything pathological about them, though?
What was that?
Whether there's pathology in the feet. I'm not aware of
I'm not
any
aware of any
foot pathology, Bob. And
Yeah, but I would come back to the point that we do not have a single postcranial skeleton of a microcephalic, and particularly with MOPD2, which is what I would want to look at. All I can say is this YouTube sequence showed that the guy concerned had strange limb proportions, and his legs looked asymmetrical and so on. So, I mean, there were clearly unusual features of the postcranial skeleton, but it wasn't possible to see what the foot was like. But they looked like overlarge feet to me.
There are other pathologies that don't involve quote-unquote microcephaly that result in small brain size or small heads, and some of those have been proposed as accounts of the peculiar morphology of Liang Bua 1. For example, it's been suggested that this was a person who suffered from goiter from an iodine deficiency, that this was a case of, what's the word I'm looking for?
Cretinism.
Yeah, cretinism. And we know a lot about the postcrania of Cretans, and I think actually the morphology of the Liang Bua specimen is not compatible with that explanation.
This is true.
And in fact, with that condition, the brain size is reduced, but it's not reduced dramatically like this. So we're talking about 900 cc's instead of 400.
well, there have been many pathological explanations posed. Were there any, Bob, that you found, or Fred, that you found even remotely convincing besides the microcephaly? Or do you think the microcephaly best explains this, or the normal species hypothesis?
Well, Gary Richards at Berkeley has suggested that it was a combined thing, that it was a genetic mutation which affected both the growth hormone axis and brain development as part of that.
Microcephaly.
Yeah. And so he wasn't disagreeing, he was just arguing there was a different genetic pathway that led to that. And that's fine, I don't see a problem with that. And it's difficult to know how, with the evidence available, we could distinguish. There was another paper from Israel which suggested that it was Laron syndrome, which is a specific condition, but that doesn't go with particularly small brains either. So I think we can forget the iodine argument and the Laron syndrome, because you just don't get this kind of small brain. You only get brains that small with microcephaly or something associated with it.
But of course, we don't know what's causing the microcephaly. I mean, you made that point earlier, but I think it's worth stressing again. It could be any number of syndromes that underlie
Absolutely.
that microcephaly.
Yeah.
Bob, if LB2, when and if we find her, looks like LB1, would you in fact abandon all explanations for pathology, given that we've got 400 genes that might produce a complex similar to
She's
this?
saying, even if you get a second individual that looks like LB1, given that there are 400 different possible and then probably more genetic syndromes that would account for this, would that rule out pathology as an explanation?
Yeah, and Fred has made this point, and I've said it too. I think the only way forward from this point is new material. If they find new fossil material on Flores, that might help to resolve the issue. And if they find a second skeleton with a skull exactly like this, then I will go back to the drawing board, because the chances of finding two individuals, which are both pathological, and not finding any normal individuals, would seem to me is stretching it. So, but if they find a skeleton which has all of the other features or some of the other features we've seen, but a skull that has the same dental features, but a brain 3 times as big, then I will.
Or even twice as big.
Or even twice as big,
Yeah,
but
I mean,
significantly larger than ours.
And there is one thing that's really, really ironic about our discussion, is that I've been more or less arguing that, you know, we ought to at least consider that this might be a separate lineage, okay, separate species. I am theoretically pretty opposed to the idea that there are any separate lineages in late human evolution. I'm one of the people who would argue that Neanderthals are not a separate species from us. And I think Bob is a little bit more predisposed to see multiple lineages, so it's kind of ironic that here I am arguing for this other lineage and Bob is arguing against it. I find that really interesting.
Exactly. I have no problem with multiple hominid species, just not this one.
Well, I have no problem with earlier multiple hominid species. I have a problem with it when humans become very generalized in their adaptive strategy with the genus Homo. That's
More questions out there?
Can I just pick up
Sure,
on
yeah.
one point, because somebody mentioned this, the pinky. It was you.
It was me.
Okay, this is a single finger bone, and Svante Pääbo was doing this survey looking for Neanderthals, and everything fits beautifully into a cluster apart from this one finger bone, which has very different DNA. It's not like Neanderthals and it's not like us, but it's human. And so when they do the tree, it gives a divergent state of, 800,000 to 1 million, something like that. And one possibility I suggest in discussing with Fred is that this is not a new hominid species either. It's late surviving Homo erectus, because the finger bone is, I think, 40,000. So it's relatively recent, but the divergence time is around 1 million. So this finger bone is a representative of a hominid lineage that survived until 40,000 years ago. And one thing that's very relevant here is on the island of Java, there is a late surviving Homo erectus population in Gandong, which has at least 6 well-preserved crania where we can get the brain size. And it's disputed whether it's Homo sapiens or late surviving Homo erectus, and you know, people diverge on this. I don't really care, but there in time and space, this is the nearest thing to the Hobbit. It's on Java, and it's just a few islands away. And whatever the Ngandong hominids are, they have pretty large brains. The average size is 1,100 cc's. And so that would be the most logical thing in time and space to derive the Hobbit on Flores. But nobody's talking about that as a possibility. And so if you believe that's late surviving Homo erectus, maybe the pinky is too.
Could be. Mm-hmm. I mean, I don't believe that Nandong is Homo erectus, but
what is it?
I think it's a part of an early post-erectus population, so it's equivalent to something like Petrolona in Europe.
Yep.
But again, I mean, that's really is not too important with vis-a-vis our arguments. But one of the things I think that reason why they haven't considered this is that people are fairly uncritically accepting the 50,000-year-old date for the Nandong material as well. And so if it's 50,000 years old, it's too recent, given what they're saying about when these guys got to.
Yeah, that's right. I mean, I'm forgetting about all that. So.
Well, I'd forget about that
I'm
date,
very
too.
sorry, but you know, there's this supposed evidence for really early tools, first 800,000 and now 1 million on Flores. These are stone tools that have clearly been transported by water,
Right.
and the authors themselves admit this. And so the question is, were they transferred into older deposits, and is the date correct or not? And this is why Fred said it's dubious. I would say it definitely is dubious. And so I'm just talking, this is what we have, and this is 18,000 years old, and I've seen values between 50,000, 53, I think 53,000 to 27,000 for.
That's correct.
Those. And you know, in time it's pretty close to this, and maybe this is a microcephalic one of those.
But Bob, if you're going to be skeptical of the soil basin dates, and I agree with you, you got to be real skeptical of the 53 to 27,000 year old dates on this, because those dates are derived from fauna that come from the same terrace as the Nandong skulls do. But if you look at them, the surface nature and configuration of the bones are completely different. These are very heavily mineralized. They're a dark chocolate brown color. Color doesn't matter, but they're very heavily mineralized. Those animal bones that were dated are very heavily leached. They're not heavily fossilized, and I don't believe there's any good reason to assume that the faunal material on which the dates for Nandong were taken are associated with those
Do you
skulls.
have a guesstimate of what the age might be?
Yeah, it's 250,000 years old, plus or minus 200,000. I don't know. I mean, I actually, I think that we're probably looking at something here in excess of 100,000 years, but maybe 100 to 200,000. But I don't know for sure. I mean, that's a guesstimate.
Fred, aren't the Nandong skulls, don't they show a lot of signs of water transport and getting
They
knocked
do.
around?
They do.
So they could be
They could
older
be.
skulls that have been redeposited in younger.
Absolutely.
Okay.
In fact, one of the things that's interesting about Nandong, and it's good you took the face off here, but the Nandong skulls, none of them.
Because that isn't Nandong.
This isn't Nandong, but Nandong does look a little bit like this. Okay, but none of the Nandong skulls have much at all of the cranial base on them. The faces are gone, and when this was first reported years ago, the argument was that this was some kind of a cult of skulls where they were opening the base of the skulls to get at the brains and all of this. Well, we now know that's not true. That this is the result of taphonomic processes that have broken the faces off and broken the cranial bases and that sort of thing. But we don't have any faces for the Nandong people for that reason. Sorry.
So, Bob, you brought up DNA, and a lot of students have come to me asking about DNA with the Hobbit, and why don't they just take the DNA? Because isn't that the solution to the problem? So why don't you guys talk about the DNA of the Hobbit?
It's the obvious way to go. I mean, both Fred and I said we need more specimens, but if we could get DNA out of this guy and be sure it actually is her DNA and not from the guy who dug it up, then it would be very interesting. Unfortunately, there is nothing published about this. It is known that a sample was taken because that was written up in Nature. And there was a big argument about it because two people from Leipzig went and took a sample, and apparently they weren't allowed to. But anyway, nobody knows what happened with that sample, so we've not heard back any results as to whether it had usable DNA or not. But as far as I know, nobody has done what I think would be the logical thing. If you want DNA, it's best to go into the pulp cavity of a tooth, because that's where it's most protected. And DNA doesn't survive in most skeletons, even in something 18,000 years old. But if you took out one of these teeth and drilled into the pulp cavity, that's probably the best chance. And I don't think anybody's doing it. And if I wanted to really be scientific about this, I would move heaven and earth to try to get a DNA sample.
Mm-hmm. Fred,
I agree.
Yeah.
I agree. I mean,
so we need to. I'm sorry to cut off.
I guess what I would caution everybody about DNA is that just because there has been DNA reported, for example, from Neanderthals, okay, that doesn't mean that every specimen that is Neanderthal age or younger we can expect to get DNA from it. It's a very unusual set of circumstances that there is organic material preserved even in the pulp cavities of teeth where you can get a DNA, where there's enough material where you can get a DNA signal. And so, you know, everybody, that's the big question today. Oh, well, why don't you just get the DNA from it? Well, that's not going to happen very much at the time. We can't depend on.
But I think
that.
the real question is, why don't you try to get DNA?
I agree with that. But I'm just saying it's not something that we all should expect to occur regularly.
Okay, well, we need to wrap up. I want to thank you guys very, very much for the conversation you just had and for the kind of civil discourse that really can happen in this field despite opposing viewpoints on things, and the agreement that more evidence is always necessary to test these hypotheses, and we shouldn't be afraid of asking these questions and challenging some of the ideas that folks have. So thank you very much for your questions and for your attendance, and we're going to be around for a little while having refreshments in the next room over there, so you're welcome to join us and ask questions of everyone on the panel. Thank you very much.